So today's lecture will be on sequence analysis, more specifically on the tools that people use to compare two DNA sequences, whether that's in the scenario where you, for example, get some short reads out of an Illumina sequencing run and want to figure out where in the genome those reads came from, or in the case where you're comparing, for example, two genomes and you want to ask questions like, how does the human genome compare overall to the structure of the mouse genome? So today, more specifically, we're going to cover two basic operations, something called a local and a global sequence alignment. And we're going to discuss how those are typically implemented and what, for example, gap penalties are and how they relate to sequence alignments. And then we're also going to briefly look at a visualization tool called dot plots, which are typically used to visualize the similarities between two different DNA sequences. So let's start with a basic pairwise sequence alignment between two sequences X and Y. So a pairwise alignment is really just a mapping between X and Y, both to kind of visualize and quantify which parts of sequence X match which parts of sequence Y. And so here in this toy visualized alignment, you can see that there's a vertical bar that connects bases in X and Y that match, or what we call a line. And so I've also highlighted in red parts of X or Y that don't match the other sequence, or these indicate what so-called mismatches in the alignment. And so there's two kinds of mismatches usually. And so, for example, if you look at the red column where there's an A in sequence X and a C in sequence Y in the middle of the alignment, this is a mismatch because the base in X doesn't match the base in Y. And so on the other hand, if you look at the other two columns that are colored in red, you can see a dashed character that we typically refer to as a gap. And so gaps in one sequence, like say in sequence Y, that match a base in sequence X, like the G, they indicate that an indel has occurred. And so assuming that, say, sequences X and Y are homologous, for example, if X is the human genome and Y is the mouse genome, indels indicate that either deletion occurred in, say, sequence Y, but not X, or an insertion happened in sequence X but not sequence Y. And so these are called indels because we can't really be sure whether the insertion or deletion event occurred. We just know that one of them happened. And so it's important to point out that there are two basic kinds of pairwise sequence alignments. First, there are those that are called global sequence alignments, which basically mean that for two input sequences X and Y, you want to align every single base in sequence X to every single base in sequence Y. And so these are obviously called global sequence alignments because you're aligning everything in X to everything in Y. And this is typically done in cases where, for example, you want to align, say, an entire genome, like the whole human genome, to the entire mouse genome to kind of get an overall view of how X really matches up to Y. And the other kind of alignment is what's known as a local sequence alignment. And so in the local sequence alignment case, not every base of every sequence has to be part of the final alignment. And so a classic example of a local sequence alignment is when you're doing, when you're sequencing a human genome using, say, short read Illumina technology, and you want to map each one of those short reads back to the entire human genome to figure out where that read came from. And so in that case, you would do a local sequence alignment because you really only care about, where are the short positions that your single read map back to the entire human genome. And for example, there may be multiple possible locations where that short read came from. And so why do we align sequences? And so it turns out that sequence alignment really forms the basis of most analysis tasks in genomics assays and genomics applications. And so, for example, in the DNA sequencing lecture, we talked a little bit about how one of the applications of DNA sequencing is to sequence the genomes of new organisms. And so we briefly touched on how it's currently not possible to sequence, for example, entire chromosomes as single reads. And so what happens is that, during the sequencing process, we are essentially sequencing short or long fragments of, for example, original chromosomes. And then we need to basically use alignment tasks. We have to use alignment algorithms in order to figure out which reads follow which other reads from the original chromosome and essentially reconstruct the original chromosome from these short and long reads. And so we're actually going to find when we get to, for example, the RNA sequencing lecture or epigenomics lecture or TF-DNA interaction lecture and so on, that basically most omic sequencing technologies amount to taking things like RNA and using protocols to convert those RNA molecules into DNA molecules, which we then feed through a DNA sequencing machine in order to generate reads that then get mapped back ultimately to the genome. And so this is useful for, for example, because in RNA sequencing, what you're doing is you're taking short fragments of RNA, you're converting it to DNA, you're sequencing it, and then the alignment process is what allows you to figure out where in the genome did that RNA originate from, or did it likely originate from. And so finally, sequence alignment also has a lot of applications in terms of studying, for example, the evolutionary history of genomes. And so in that kind of application, you might, for example, align 2 or more genomes together to then figure out what kind of events might have occurred since the common ancestor of those pair of genomes. And so you might try to infer, when did certain types of deletions or insertion events occur, and so on and so on. And those kind of tasks basically all involve, at the core, doing sequence alignments between pairs or more of genomes. So it's worth pointing out that between any given pair of sequences, there's many possible alignments that you can have between them. And so the alignments that we tend to look at and that tend to be the most useful are the alignments that yield the best score, the smallest edge distance. And so what that means is that the best alignment between a pair of sequences is the one that yields the smallest number of differences between those sequences. And so here, for example, are 2 examples of alignments between the same pair of sequences, X and Y. And you can see that the alignment A implies a smaller distance between the pair of sequences because there's only 2 differences implied by that alignment. Whereas in alignment B, there's actually 3 differences implied. And so again, if you want to kind of since sequence alignments are typically used to identify the relationship between 2 input sequences, X and Y, then the alignment to the smallest edge distance tends to be the most informative. So figuring out which alignment is the best between a pair of sequences can actually be a little bit more difficult than just calculating the number of matches or mismatches implied by each given alignment. So if you consider this slide, for example, if you compare alignment A and B, those 2 alignments are of the same input sequences, X and Y. And it's pretty obvious from comparing alignment A and B that alignment A is better because in both cases you have 3 gaps. And for alignment A, you have 3 matches, and alignment B, you only have 1 match. And so in this case, alignment A is clearly better than B in terms of minimizing the number of differences between the 2 sequences. But if you look at the second example, comparing alignment C and D, it's a little bit more complicated to figure out which alignment is the best because in both cases, you have 3 matches between the 2 sequences. But if you look more carefully in alignment C, you can see that there's a gap implied between the A and the CC in the bottom sequence. Whereas in alignment D, you don't have any gaps that are interior to the bottom sequence. And so if you think about the comparison of the 2 sequences from an evolutionary perspective, alignment C basically implies that there was some kind of indel event between the A and the CC, whereas there's no indel event implied in alignment D. And so intuitively, alignment C is probably a worse alignment because it implies that there was an indel event, which tend to be rare in terms of genomic events, compared to alignment D, where you just have one sequence that's shorter than the other. So an important point that I want to make here is that one of the primary goals of sequence alignment historically anyways is to basically help identify homologous sequences. So one of the first applications of sequence alignment was really this idea that you sequence a new genome or a new gene, and you want to find homologous copies of that gene in other organisms. And so you would use sequence alignment to find other sequences in a database of gene sequences other homologous copies of that gene. And so if your goal is to, for example, give high scores to pairs of pairs of sequences that are homologous, then one way that you should do that is by making scoring schemes that tend to reflect how genomes evolve. And so what I mean by that is, for example, transition. So when you talk about genome evolution, transitions, at least in humans anyways, transitions tend to occur more frequently than transversions. And so if you want an alignment that captures that notion, then you should essentially penalize transversions more highly than transitions, because they do tend to be more rare, and so they shouldn't happen in sequence alignments as often. Similarly, in terms of genetic variation across populations like humans, you tend to get substitutions, or basically what look like mismatches in your alignment, rather than indels, which produce gaps. And so real world alignment scoring schemes will tend to penalize introduction of gaps more than, for example, just mismatches in an alignment. And finally, it's also worth noting that for DNA sequencing technologies, usually mismatches or substitutions tend to happen more frequently, or sorry, not substitutions. Sequencing errors tend to lead to single base pair changes rather than introduction of gaps. And so gaps are typically more penalized than mismatches for that reason also. And so here's an example of a more realistic scoring scheme for an alignment used in real life. And so here, for example, like larger values mean stronger penalization. And so here, for example, transitions again are penalized not as badly as transversions. And so it's worth pointing out, again, going back to the idea of gaps, and gaps generally being uncommon in evolution, or less common than substitutions. In real world scoring schemes for alignments, people typically actually distinguish the idea of a gap opening in an alignment from a gap extension. And so the intuition here is that introducing a gap into an alignment is bad, because indels typically happen less frequently than substitutions. But having a gap of, say, length 50 bases is not actually functionally worse or much worse than having a gap of size 51, right? And so typical scoring schemes, when they look in alignment, they penalize a gap by saying, okay, if I have a gap in a particular region of my alignment in the first place, then I'm going to penalize that with some penalty called G0 in this case, or basically an opening gap cost. And then I'll penalize the existence of a gap, and then I'll penalize the gap also in terms of like how long that gap is. So if the gap is only like 2 bases, then I'll penalize it 2 times the cost of G1, which is the cost of extending a gap by 1 gap character. And so the total cost that is assigned to a given gap in alignment is basically just this G0 penalty plus G1 times however long or however many gap characters are introduced in that particular gap. And so generally speaking, this G0, or cost of opening a gap, is typically much higher, of course, than, for example, G1, to reflect the fact that having a gap is bad, but extending it is not typically as bad as opening one. So up to now, we've been mainly focused on the task of pairwise global sequence alignment, where we have 2 sequences, X and Y, and we're just trying to find the best alignment of basically all the bases of X to all the bases of Y. We won't really delve into this topic any further, but there does exist a type of alignment called the local alignment, where essentially, suppose that you have, for example, 2 genomes that have diverged like hundreds of millions of years apart. And so suppose there's just very, very little sequence identity globally between 2 genomes. This is one example of a case where you might want a local alignment where you might presume that there exists some smaller segments of your genome, say individual genes that you think are highly conserved between a pair of species, but you think globally the two genomes are very different. And so what a local alignment does is it searches for best matches between a pair of sequences X and Y, but they allow those best matches to be only parts of the genome, only parts of either genome actually. And so local alignments really strive to just find matches that are possibly just short matches between pairs of genomes, and they allow for more than one partial match essentially. And so in this diagram here that I'm illustrating, I'm just showing you one hypothetical example where the best match, the highest scoring alignment between a pair of sequences X and Y is just made up of a smaller, basically subsequence from X matched to a smaller subsequence of Y. But we don't really need to care about the details of local alignment in this particular class. One other situation in which doing local alignments may make sense beyond identifying small conserved gene sequences between genomes is if you have, for example, you're comparing two genomes, but say one of the genomes has undergone, say, rearrangement. And so pairwise global alignments, for example, can't do a very good job detecting inversions normally, for example. And so earlier on in the lecture, I mentioned that one use case of sequence alignments is to identify evolutionary relationships between those sequences. And so, for example, you might align the human and the mouse genome in order to identify homologous regions between these two genomes. And so a question that oftentimes gets asked is, if a pair of sequences achieves a higher and higher alignment score, does that make them more likely to be homologous? And the answer is that not necessarily, because of processes like, for example, convergent evolution. And so in convergent evolution, you might have, for example, two different protein sequences that, because of certain external constraints on the function of those proteins, basically encourages those protein sequences to look more and more similar, because sequence similarity implies functional similarity. And so you can have two sequences that achieve relatively high score, not because they shared the same origin, but because they were required to have functional similarity. And so it's also worth pointing out that for a given pair of sequences, even with functional constraints, sequences basically can diverge over time, because of lack of selection or other processes. And so basically, if you are comparing, or if you're aligning a pair of sequences from two different genomes, depending on where in the genome those pair of sequences come from, some sequences may evolve faster than others. And so that's another way of saying that even when you have pairs of sequences that are truly homologous from the same pairs of genomes, their expected similarity in sequences, identified by the alignment, can be different depending on what the role of those sequences had in terms of the genome. So a pretty common tool for helping visualize sequence alignments, and in particular genome sequence alignments, is the dot plot. And so the idea of a dot plot is shown on the right, where it's basically a heat map of where the rows are the different positions of sequence 1, and the columns represent the different positions of sequence 2. And so the idea of a dot plot is that basically, if you consider the cell in this heat map at row I and column J, so that's what I'm calling Sij here. Basically, there's only two possible values for Sij. Sij is either 1, which happens if if a short sequence around position I in sequence 1 and the short sequence around position J in sequence 2 have a high alignment score, so if they match pretty well, essentially, then the score or the heat map at that position ij is 1. Otherwise, it's 0. And so in the heat map on the right, basically blue means 0, so there's no strong similarity between most positions in sequence 1 and position 2, and sequence 2. But basically where you see the white lines, that's where you have high sequence similarity between position I and J across sequences 1 and 2. And so if you look at the heat map on the right, what you'll see kind of like two diagonal lines. And what those lines represent is basically that the start and the end of sequence 1 and 2 basically match pretty closely. What you see though is you see kind of like a vertical gap in the heat map, and that corresponds to basically a deletion. That tells you that essentially there's a deletion in sequence 2 relative to sequence 1. How you can kind of infer that is that if you look across the different columns of this heat map, you'll see that for every column in this heat map, which corresponds to a position in sequence 2, there's a white box in every column of this heat map. And so what that tells you is that every position in sequence 2 has some matching position to sequence 1, but the reverse is not true. If you look at all the rows across this heat map, you'll see that in that kind of gap area that I've circled with a red oval, there is basically a segment of sequence 1 for which there's no matching position in sequence 2. And so that again tells you that there's either an insertion in sequence 1 or deletion in sequence 2. And so here's an example of a dot plot between two sequences that are identical, except for an insertion of 3 bases in the sequence represented by the columns. And so I've colored those 3 bases in red just to make them easier to see. And so the idea is that when you want to draw a dot plot, really what you're trying to do is you're trying to consider every base in the first sequence, which is represented by a column, against every base in the other sequence, which in this case is represented by the rows. And so for each column and each row between the two sequences, which is basically represented by a single box in this table, you want to decide whether or not the sequence centered around that column and that row is similar enough such that you want to put a dot there to indicate that, you know, the local similarity between the two sequences at those positions is high. And so here, for example, I've drawn an arrow to one row and an arrow to one column to represent the 2 bases that I'll consider putting a dot there to indicate high sequence similarity. And so when you draw a dot plot, you have to decide 3 different parameters. The first parameter is basically the window size, which says that, okay, you know, given that I'm looking at, for example, the column that's pointed to by the arrow, how big of a window around that column do I want to consider as my local sequence? And so here in this example, we'll select a window size of 3, which means that when I'm looking at the column represented by the, or that's being pointed to by the arrow, what I'm considering is my local sequence is basically that column, that T, as well as the one base to the left and to the right of that. So really I'm considering the short sequence ATG represented by the first 3 columns. And similarly with a window size of 3, then I'm just really looking at the first 3 rows of the second sequence. And so when I decide whether or not to draw a dot at that row and column indicated by the arrows, I'm really asking the question, is ATG similar to ATG? And so the first thing you need to do is, once you've defined your windows and defined the row and column that you're looking at, you want to basically just compute how similar are those 2 short sequences. And so in this specific example, we'll just give a score of +1 if the bases match and say -1 if they don't match. And so because ATG and ATG are identical, that means that their score is 3. And so finally, you have to also decide a threshold. And so the threshold basically just says if the score of the matching between these 2 short sequences is at least, you know, the threshold amount, then draw a dot there. And so if I set the threshold to be 3, then these 2 windows actually match up and they pass the threshold because they're a perfect match, and so they get a score of 3. And because 3 is at least as big as 3, then I'm going to put a dot at the row and column pointed to by the 2 arrows. And so I can basically you want to go through this thought process for every row against every column in this entire table. And so because in this kind of made up example, sequence 1 matches sequence 2 exactly except for an insertion, basically what you'll see is you'll see more or less two diagonals on this dot plot. But one of the most important features of this dot plot are that you'll notice that for the 3 bases that correspond to the insertion in sequence 1, you'll notice that there's no dot in those corresponding columns. And that's because the insertion in sequence 1 doesn't have any matching sequence in sequence 2. And so that's why you don't see any dots in those 3 columns. On the other hand, you'll notice if you look along the rows, almost every row has a dot in it, with the exception of just a few rows around the boundary of the insertion. But basically, the point of this dot plot example is to show you that if you have 2 sequences where they only differ by an insertion, it's easy to see that from the dot plot because you'll see that in this case, you'll see a whole bunch of columns missing a dot, but most of the rows have a dot. And so what that means is that there's some part of sequence 1 that doesn't have any match to sequence 2, but every part of sequence 2 has a match to sequence 1. So that's how you can automatically tell that there's an insertion in sequence 1. And so here's an example of a question that, I want you to that I think you should take a few minutes to answer before you move on to the next slide. So I think, now is a good time to take like a 2-minute break to essentially think about what this particular dot plot tells you, right? And so again, just to give you a hint, the diagonal lines on at the beginning and the end of sequence 1 and 2 basically tell you that the start and end of sequence 1 and 2 match pretty closely. But I want you to think about I want you to stop the video and think about what that kind of off diagonal line that I boxed in dashed lines represents. So here I'm drawing another example dot plot, where in this case I'm showing what a dot plot would look like if there is a single duplication event. So suppose we have 2 genome sequences, one corresponding to an ancestral genome, so that's the genome sequence of an organism that we don't observe today. So like, for example, the human mouse ancestor. And suppose we're dot plotting that ancestral genome against a so-called extant genome. So that an extant genome is basically a genome of an organism that we do observe today. So say, for example, humans. And so suppose that in the extant genome, we observed a single duplication illustrated by the colored boxes on the rows of this diagram. And so what you notice here is that when there is a duplication in this case, the extant genome, what you see in the dot plot is that every row of this dot plot, so that corresponds to every position of the extant genome, has only one dot in every row of the dot plot. And that's because there's an exact match to some position in the ancestral genome. But on the other hand, if you consider the columns of this dot plot, which correspond to positions of the ancestral genome, there's actually 2 dots in every column that was duplicated. And so the point here is that when there's a duplication event for the shorter sequence, you'll notice that there's kind of parallel lines in this diagram. And so that tells you where the location of the duplication event is. And so here's an actual alignment between the human and chimp genomes. So these are whole genome alignments between human and chimp. And so what the vertical lines in this dot plot represent are the boundaries between the different chromosomes on the human genome, and the row and the horizontal lines across the rows basically give you the boundaries of the chimp chromosomes. And so what you can see is, again, as you'd expect, because human and chimps are the genomes are relatively similar, you see a lot of bands along the diagonal of this entire plot, which says that there's a lot of large-scale matches between the human and chimp genomes, which is what you'd expect. But what you also see is a bunch of off-diagonal lines. And so I've indicated here, what's indicated here is basically an interpretation of what those bands tell you. And so I also, again, kind of want you to think about, I want you to look at these bands and try to understand, okay, try to convince yourself why an inversion looks like the way it does, or a relocation looks the way it does. I've shown you here on the right 2 additional heat maps, which kind of zoom in on the dot plots for the Y chromosome, specifically in chromosome 21. And this is just to illustrate again that for some chromosomes like chromosome 21, basically the plot looks more or less like the human and the chimp. Chromosome 21 matches pretty well. Whereas for some chromosomes like chromosome Y, you see a lot of kind of small to mid-scale genome rearrangements happening, as evidenced by the off-diagonal lines. And so again, just to summarize, for the purposes of this class anyways, what we do with these alignments is mainly we use alignments to map reads to the genome. And so, for example, here's a plot showing you reads from an RNA sequencing experiment that had been mapped to known coding sequences on the genome. And so basically the point here is that each of these short little lines corresponds to one read that you've sequenced. And so basically the goal of this visualization is to really just kind of see, okay, where do all my reads pile up? Essentially, where do I see a high concentration of reads mapping or aligning to a particular region in the genome? Because basically the more reads that I see mapping to a given region, that means that for RNA sequencing experiments, that's more indication that gene's being expressed or that transcript's being expressed. For other experiments, like for epigenomic assays, those reads tell you where potential like enhancers or regulatory elements are. And so basically where these reads pile up gives you some evidence of some kind of, maybe not perhaps function, but there's at least some, for example, in this case, expression happening at that locus in the genome. And so sequence alignment, in terms of the kinds of software that you use to align reads to the genome is a pretty mature. The software is pretty mature. The 2 to 3 kind of main challenges that might be relevant even today is number 1, speed. And so obviously, depending on how much sequencing data you're producing, a lot of these kind of alignments, especially when you talk about genome scale alignments, can't really be done on your computer, not very easily anyways. And so in practice, if you were to, try to go home and, if you got really excited about doing genome alignments and went home and tried to align the human and chimp genome, you might not be able to do it on your laptop. You'd oftentimes have to use like a high performance computer to do this in practice. And especially for people that do a lot of like metagenomic sequencing and things like this, speed starts to become a problem because you can really generate really large scale data sets, especially with a lot of those like portable sequencing machines that are available today. Another main challenge is that really alignment, at least of reads to the genome, which is the main application of alignments that we talk about in this class. One of the main problems you have are basically the question of, are there really good reference genomes available? And so for organisms like humans or like, Drosophila or C. elegans or yeast, there's a lot of relatively good reference genomes available. But if you're studying things like, if you're doing things like metagenomics or the microbiome or things like this, there oftentimes is a lack of good reference genomes available. And so that's where a lot of problems with alignment happen. And actually, specifically in the RNA sequencing lecture, we'll talk about problems related specifically to aligning short reads. And so again, the problem of having good reference genomes available becomes more and more important as you have the shorter your reads are. And more specifically, especially when it comes to RNA sequencing or Hi-C, which we'll explain later, split reads become a problem. And so I mentioned earlier that in the review lecture that obviously alternative splicing is a pretty prevalent occurrence in like higher eukaryotes. And so the problem you have is that suppose that you have like a really highly alternatively spliced gene, where some of your exons that get spliced together are actually pretty far away on the chromosome. But suppose you don't know what the structure of your genome is ahead of time. The problem you have is that if you're trying to, for example, detect alternative splicing, alternative splicing with short reads is a huge problem, because if when you're trying to characterize alternative splicing with RNA sequencing, and you basically want reads that map over junctions between exons, because reads that span single reads that span multiple exons tell you which exons get spliced together. But if you are trying to, if you have a short read, say like 35 base pairs from Illumina sequencing, and that 35 base pairs spans like a junction between two exons that are like really far apart on the genome, then the real alignment that will tell you that those two exons that are really far away on the genome get spliced together, the real alignment there will have could have like tens of thousands of gaps corresponding to the sequence in between the two exons. And so you could imagine that under any like real scoring penalty scheme, the score of the real alignment will be like really bad. And so in the RNA sequencing lecture, we'll talk about strategies for getting around that problem.